Chomsky, nothing makes sense in biology except in the light of evolution. Here's a more recent sort of mock quote by Michael Lynch, a guy who we'll be talking about a lot, rather large, big population geneticist of nowadays, our contemporary, saying that nothing makes sense in evolution except in the light of population genetics. And again, the reason why this is true is that population genetics actually gives us rather basic models to understand what, for example, the dynamics of allele frequency in a population should be under rather simple forces and then under their combinations. And the predictions it makes, population genetics makes, are sometimes rather non-intuitive. The fact that some of us fall ill, the fact that the fitness of the population is not optimal, is not maximized, the fact that the population properties should depend on the population size, none of these things make sense immediately, but they will make sense after you study population genetics and look at the equations. So this is something I think that is critical to understand for any biologist, and that's something we'll be doing for the next couple of lectures. So again, what exactly is population genetics study? And we have discussed how the macroevolution deals with the movement in the space of traits. There's this hugely multidimensional space of traits, and we are trying over the course of millions of years to get from one point in this space to another, and we study the paths in this multidimensional space. And there can be a whole lot of things happening, and we can be traversing this space in many different directions, and a lot of different traits can be affected, and a lot of different alleles can spread or get lost along the way. Population genetics tries to simplify this incredibly complex picture by looking at something very simple, and the simplest thing we can come up with is a binary trait with just two alleles, 0 and 1. And what we are trying then to study is, yes, so here, just one more. Here we have been analyzing this whole complex movement in this genotype space, but what we have been sacrificing is the variability within the population. We have been just looking at the species as represented by a dot and assume that all the individuals are identical. What we do here is we look at a single trait coded by 0 and 1, but we are accounting for the variability in the population, and the simplest thing we want to understand is the dynamics of the frequency of this allele in the population. So the space we are dealing with is this line between 0 and 1, and that corresponds to the two frequencies of the allele we're considering in the population. Pretty straightforward. The simplest thing we can be thinking of. So we're moving in the space of trait frequencies, here one-dimensional, although of course we can look at combinations of different traits. Actually, we'll have to do that when we start talking about how the traits interact with each other in the context of sexual reproduction and also in the context of epistatic interaction between traits. But for now, let's just focus on this one-dimensional picture. So we're moving in the space of trait frequencies, and we're dealing with microevolution. And the science that tells us the laws under which this happens is called population genetics, and this science has been really well developed over the course of the 20th century. And somewhat similar to some areas in physics, the people wrote the equations actually before there was data to support the equations, and before many of the parameters that were important in those equations could be actually measured. So people instead used some guesstimates for these parameter values, such as the genome-wide or locus-wide mutation rate, such as the distribution of selection coefficients, such as the effective population sizes. And now is a very exciting time to do population genetics because we can actually directly measure these parameters by studying the genomes of genotypes of, by now, tens of thousands or hundreds of thousands of individuals, for example, of our own species, but also of other species. Fine. And so we're talking about the frequency of an allele in a population, and we have discussed in some depth what population is. But basically, just to remind you, the population is, we will be using this ad hoc definition of population as the set of individuals that can be conveniently looked at together in the context of the task at hand. And the reason for this informal definition is that we might want to have different definitions for what a population is depending on the time scale we want to consider. And just to remind you, we have this example. There are two forests, and there are bears living in this forest and bears living in this forest. So there are bears here and bears here, and they live independently, but once every 100 generations, there is a bear going from one forest to the other, and then getting involved in the production in the other forest. So is that one population or is that two populations? The answer depends on the question we want to ask. If we are interested in the negative selection against deleterious mutations within the populations, then we don't really care about these rare wandering bears. Instead, we can consider these populations independently because the allele frequencies will be largely independent of them. However, if we're interested in spread of advantageous mutations, this bear can bring with it an advantageous mutation which has a reason in this population, and that mutation can then spread here, and these would be very much affected by the link between these populations. Otherwise, this other population would have had to wait till this mutation arises here de novo, and so in that context, it's convenient to consider that as a single population. So the operational definition we'll be using is this one on top, and it deals with the set of competing lineages, so that an expansion of one lineage leads to the decline of others. The reason for that, and we sort of had a hint of that, I think, in one when we discussed the papers from the long-term evolution experiment, and there we said we had a population splitting into two independent populations, so that novel allele replacements in one of those subspecies, you can say, emergent subspecies, spread independently of the other. Great. Okay, so there's a lot of variation, and that's something that we need to explain, but first, of course, we need to describe somehow. So we need to come up with some description of the variability, and mostly will be, since this is a genomics-oriented course, we'll be mostly interested in the variation at the genetic level. However, of course, it is interesting also to describe the variation at the phenotypic level, and here we are immediately running into trouble because some of the traits are pretty straightforward, some traits are pretty binary, can be described by 1 or 0. Some traits can be described quantitatively by a number, such as human height, or perhaps the skin color by comparing to some predefined palette, or perhaps the body mass index. But some other traits are really hard to describe, such as the overall body shape. and we will need to have to come up with some way to describe such complex traits and the variability. And at the genetic level, the situation is in a way somewhat simpler, because we do have this discrete text consisting of individual letters, and the variation here repeats the patterns, the different patterns. The variation sort of repeat the patterns in the mutation. So the mutations, as we've discussed, can be small scale, and those can be single nucleotide substitutions, such as T2C change here, insertions or deletions, or complex events, for example, multiple simultaneous mutations at different parts of the different letters, different nucleotides at the time, or they can be structural large events. And the variation is similar in that it repeats this same classification. You can have small-scale variance, that is differences between individuals within populations, for example, humans, between 1 and 1,000 nucleotides, for example. The most prominent and famous of these are single nucleotide polymorphisms, or SNPs for short. So these are just nucleotides that are different between different individuals in populations. You can have polymorphic indels, sometimes called CNVs for copy number variants, although this is a horrible name. And you can have complex variants, for example, some individuals having a specific combination of nucleotides at some stretch of DNA, but the others having a different combination. And again, you can have these structural variants, and similarly to what we have discussed about mutations, SNPs are the most frequent type of polymorphism, so the most usual type of variation is just these changes affecting just a single letter. However, by the amount of nucleotides affected, these larger variants can affect a comparable number of nucleotides. For example, if there is a chromosomal anomaly, such as a loss of or an excess copy of the 21st chromosome, or a duplication of a segment of chromosome, which can even be asymptomatic sometimes, that simultaneously affects a huge number of nucleotides, more than that. An individual carrying it will have more differences, more different nucleotides in the genome from the rest of the population in this segment than it will have SNPs in the entire genome. Okay, so we'll need to come up with some measure of variation, and we will be primarily focusing on SNPs throughout. That's what we'll be talking most about. So how do we describe the SNPs, the variation of population in its SNPs? The problem is as follows. You have this alignment, but now it does not correspond to many different species that have diverged in the course of evolution. Rather, it corresponds to many individuals within the same species, within the same population that have had a common ancestor some time ago, but have accumulated independent mutations since then. So the most straightforward way to measure the amount of variation would be just to count the SNPs. How many SNPs are there in this first scenario? 4, right? So these are 4 variable columns in this alignment. And here,
3.
Here there's, okay. So this is a bad measure, obviously. Why is this a bad measure? For many purposes of description of variation, if you want to understand what the variation of a population is, for example, compare different species with each other or different populations with each other, this would be a lousy measure. It does not account for frequency. You know, so why would this be a problem?
Because it's possible that we have different frequency of alleles in diverse populations.
Right. So basically, it will also depend on the population or on the sample size. Right. The more individuals we include in our sample, the larger number of SNPs we will observe. If we have just 2 individuals, we will see 1 SNP out of 1,000, and if we have huge numbers, we will see huge samples. We will see some individuals that have changes in the nucleotides, even those nucleotides that are conserved throughout most of the population. And the number of SNPs grows with the sample size logarithmically. And when I started doing genomics, we just had, if we were lucky, a handful of personal human genomes. So the data set for comparison was like about 5 human genomes. We had the genome of James Watson, the genome of Craig Venter, a single representative genome of Han Chinese, a couple of genomes from Africa perhaps. So given that, we had variable SNPs in about 1 out of 1,000 nucleotide positions. Now we have tens of thousands of complete human genomes, and the frequency of SNPs is about, I believe, 1 out of 5. So 1 out of 5 positions in the genome has a change at least in 1 individual in these huge samples. Of course, the vast majority of these SNPs are very, very rare. We'll talk about that in a second. So we want to have a measure that would be independent of the sample size. One such measure, and the one most frequently used, is called virtual heterozygosity, and this is just the probability that the 2 randomly sampled individuals, 2 individuals randomly sampled from a population will have different nucleotides at a particular position. Okay? So the virtual heterozygosity, say for this 3rd position here, is the probability that we randomly sample 2 genotypes again here, and they are different. So we can sample this 2nd and this 3rd genotype, that will be identical, but we can sample the 1st one and the 3rd one and the 4th one, and they will be different. So it is easy to see why this is called virtual heterozygosity, right? Because it is the same thing as heterozygosity, that is the probability to have a heterozygote at this position, if our population is panmictic, if our population breeds randomly. So then that will be exactly the same thing. However, we can define virtual heterozygosity for populations that are not panmictic, for populations that are, for example, subdivided, or even for populations that are asexual, that do not have sex, such as bacteria. We can measure the virtual heterozygosity for bacteria, for example, that do not engage in sex all that much. And this is arguably a better measure, and this is the measure we have. We can write equations for somewhat conveniently. We can we'll be discussing what it should be equal to under a certain flow of mutations into the population, under a certain population size, and so forth. And of course, it is normally monotonically associated with the number of SNPs. So in these 2 particular instances, it is easy to check it if you like. It's nice exercise, that the virtual heterozygosity is the same even though the number of SNPs is different. So the virtual heterozygosity is both a function of the number of SNPs in the genotype and of their frequency. So here there are fewer SNPs, but each such SNP has a higher frequency, giving rise to a higher virtual heterozygosity. The virtual heterozygosity for a certain SNP is maximized when its frequency is 1/2. That's something that's easy to see. So the complete description, yes. So okay. So I've told you what the virtual heterozygosity for a position is, but the complete description should involve measuring virtual heterozygosity for a long sequence, and that's just the average of these values over all positions. And to get the complete description, this still does not tell us everything, right? This still does not tell us what the allele frequencies are, for example, because again, these two situations are exactly the same in terms of virtual heterozygosity, but the allele frequencies in them are relatively different. Here we deal with rare alleles, and here we deal with more frequent alleles. So if we want the complete picture, more complete picture, we might want to construct the allele frequency spectrum. And here is one way to do this. So here's along the horizontal axis, you plot the frequency bin in which the minor allele falls. And along the vertical axis, you put the fraction of such loci, so this is the distribution. Now what is it? This is the minor allele frequency. The minor allele is just one of the two alleles that is more rare in the population. Here, the minor allele is C and T is the major allele. Here, either of these alleles can be chosen as the minor or the major. And so this horizontal axis goes all the way up to what frequency? What can be the maximum minor allele frequency in a population? The highest? 0.5 again. So it does not go all the way to 1, it can only go up to 0.5. And this will normally look like a diminishing distribution. Most of the alleles will tend to be low frequency, and the higher the frequency of the allele, the fewer such alleles. We will see the reason for that shape of the distribution later. It arises pretty much even without any effect of natural selection, but selection makes this pattern even more regular. So here you can plot this distribution in red, the expected distribution, what it just should look like in general, but then you can see that for synonymous substitutions, it is sort of biased to the left, and for the non-synonymous substitutions, it's biased to the left even more, so that the rare alleles are overrepresented and the frequent alleles are underrepresented. So does anyone have an idea why the non-synonymous alleles tend to be more rare in the population than synonymous ones? Perhaps someone other than you then. Why are non-synonymous alleles in the population more rare than synonymous ones? Any guesses?
Because non-synonymous may correspond to some disease which corresponds to low fitness. And
right.
Because of low fitness, we have low frequency.
Right. Does everyone see what he said? So selection probably acts against some of these non-synonymous alleles, pushing their frequency downwards, reducing their frequency. Just as a side note, sometimes people, instead of the minor allele frequency, draw something called derived allele frequency, DAF. And this is a frequency not of the more rare variant, but of the variant which is derived, that is, which is not the ancestral one. We can think of this polymorphism as a stage in evolution in which we get a new mutation and then it can either spread through the population or get lost. And then we can tell here, for example, C and T, we can work, well, let's look here, C versus T, we can ask which of these two variants is the ancestral and which of them is the derived. How would you go about testing these, for example, in humans? How do you know whether C came first or T came first? Any guesses? So we have this polymorphism, right? Lots of individuals in humans that all have some common ancestor, and we have some of these individuals have nucleotide C. A bunch of guys have C, and the rest have T. How do we know whether it was originally a mutation from C to T or a mutation from T to C? Is there any additional source of data that we can use to understand that?
We can see some historical data, like saved blood from.
Like what?
Saved blood from kings, for example.
Yeah, saved blood. Yeah, if we can have access to ancient DNA, that would help. But many kings would be nested somewhere here. So the last common ancestor of all humans would have lived more anciently than all the kings. So the kings will not necessarily give us the ancestral state. Just hold on a second. Just let someone else give it a try. Perhaps someone other than you two. Wait a second. Anyone else? Go ahead.
Some relative species.
Some relative species. What's the closest species we can use?
Chimpanzee.
Chimpanzee is the closest species to human, and so that's what people usually do. If they have a T here in chimp, then they assume that T is the ancestral state, right? If this polymorphism is also, so if this T is observed in some of the humans, but C is observed in some other humans, then people think this was a T to C mutation, not a C to T. And then you can look you can look on all the alleles in the genome and plot the derived allele frequency. And that, of course, does not have to be limited by 0.5. This can go all the way up to 1. Because sometimes you can have a situation in which the allele that is derived, such as C, has reached a high frequency in contemporary humans, even though some T's remained. Otherwise, if C has been fixed in all humans, then this would not be a SNP, right? This would be just a fixed variant observed in all humans. But if C are observed in some humans, in most humans, but T in some of them, then you can get high frequencies here. And the normal distribution that you see in cases like this is something like first it goes down, but then it goes a little bit up in the end. And again, we'll probably discuss some reasons for that. Words. Does this make sense? Okay, so then I told you that virtual heterozygosity is a good measure because it is independent of the sample size, and so it allows us to do nice things such as, for example, compare different species with each other. And in our own species is rather monomorphic. We all look rather different to each other, but in fact, we're genetically much more similar than most other species, I would say. So our polymorphism level is 10 to the minus third. Let's compare that to some other species. So in mouse, it will be about a 10-fold higher. Two randomly picked mice will differ, two randomly picked mouse genotypes will differ from each other in 1 out of 100 nucleotides. About the same for Drosophila, about the same for C. elegans. So there are some outliers, species which are highly variable, and in particular in our lab we are very interested in those. The previous record holder was the marine ascidian called Cyona savigny, and for it, the polymorphism level has been 8.08. So for comparison, I've told you that the divergence between human and chimpanzee is 1%. So that two individuals of Cyona savigny is about 8 times more different than a human is to a chimp. In fact, the human macaque distance, human chimp is about 1%, and human macaque is about 5%. So these are even more, two individuals are even more distant than a human and a macaque from each other. And a human and macaque probably will not be able to produce a fertile offspring if they engage in sex. However, these Cyona savigny certainly are the same species. They certainly are giving rise to healthy offspring.
When these numbers are calculated, are these polymorphisms from across the whole genome or just exome?
Whole genome. This is whole genome data. These guys have rather short genomes. So we were really interested in looking at very highly polymorphic species because for some things, it is really great to have a high polymorphism level. For example, if you are interested in associations between different alleles, you really want to have a highly polymorphic species. And we in our lab were initially thinking that maybe, so this thing is a marine ascidian, one living in sea, and we're thinking that maybe the marine invertebrates are the ones that tend to be the most variable, just because the sea is so huge and there are so many of those. However, it seems that this is not a general trend. Instead, the most variable species that we were able to find is something called Schizophyllum commune. That's a fungus that grows all over the globe. Actually, if you go outside here on some old oak trees, probably you will be able to find some of those. So just it's a fungus covering the trees, a basidium mycete, and its polymorphism level is somewhat unbelievable, 20%. So the two individuals are really, from the same species, are different in 1 out of 5 nucleotides. And actually we're extremely excited about this thing. Sasha Bezmenova here, among others, her projects studies the experimental evolution in this fungus. And these, we have previously found that this high vertical heterozygosity comes from a number of factors, including the high mutation rate, but not just mutation rate, not just the high mutation rate. Okay, so the point is that different species really hugely differ in their vertical heterozygosity, and you can do different genetic things with them depending on how heterozygous they are. Our own species is rather monomorphic, and that complicates things if we want, for example, to look for genetic associations with human traits, such as disease. Okay, so that much about variation. Now, I've promised you that we'll be talking about how the species, how the allele frequencies change in the course of the lifetime of population. Let's make perhaps 5-minute break, even though it's a little bit early, and after that we'll dig into